Hi, I'm Jonathan.
And I'm Thomas, and we will be presenting on microscopy in biology. In a lot of cases, microscopes can be used to see much smaller things, and in the case of biology, we can use microscopes to see things such as cells, which make up the human body. This can not only help us understand more about cells themselves, but microscopes can have even more practical applications. For example, research on multiphoton microscopy, which can be used to help discover traces of ovarian, esophageal, and other gastrointestinal cancers, is done at the U of A. Microscopy is used as a tool in almost all biological studies, ranging from inspection of cellular detail for tissues to fluorescent dyes that bind to specific proteins or other targets. It is also commonly used for studying live cell cultures.
So focal length is pretty much the distance that light is focused from a lens, right? So light enters into this lens and it's focused right there, and that's the focal point. This is the focal length. So here we have a model of our microscope, and this is basically the path of light that goes through our system. So in our microscope, the ratio of the focal length between the objective lens and the eyepiece lenses, focal length combined, is what determines the magnification power of our microscope. And you can see here with the whole distance, as the objective lens focal length decreases, or as it gets smaller and smaller, right, this slope would get more steep, right, because the focal point would be closer. And this would ultimately result in more magnification because this slope would essentially mean the light is getting focused more and more highly in one spot in the eye, right? Or whatever detector you have.
Here we have a programmed model of the system. As you can see, with the smaller focal length objective lenses, the final image gets more spread out. This is indicative of the magnification of the microscope. Here is the first design for the mount to hold the lenses. It will hold all three lenses in place. Our second design holds a single non-mounted lens. Here we have the same setup as before, but with our other design of each lens in its own holder. With this setup, we have to make sure that the lenses are aligned, so we have them pushed up against the book here. We started with the same distances between the lenses as before, but now with this setup, we can separate the lenses and we can see the effects that it has whenever we change the distances between the different lenses. Good.
Alright, so first we will make the eyepiece by placing two 25mm lenses in the two sockets farthest from each other. And then we'll take the 75mm lens first and put it in the objective lens slot. And then what we can do after that is we can put a dollar bill there. And then you put your phone up to the lens, right? And then you can see as you get it to the right point, it's focused right on the dollar bill, and you can get zoomed in on it just like you would with a normal microscope. And right here you have a simple microscope with a 75mm objective lens. Now, what you can do after this is you can switch out your 75mm for the 50mm. And you can put your phone there again, and you can see there's a bit of a difference, right? It looks clear, it's a little bit more zoomed in. And then after that, maybe taking a picture, you switch out your 50mm for your 25mm lens. Make sure it's all lined up. Put your phone up to it, and you can see you get even more zoomed in.
Here are the images we took through our compound microscope of a $1 bill. You can clearly see that the magnification is much larger with the smaller focal length objective lenses. Looking at the letters on the bill also makes it easy to see the inversion of the image.